Okay. Okay. 44 here. Cool. All right, off of gram-positive stuff. Gram-negatives now. Serratia marcescens. So Serratia, this is kind of a really cool bug in the sense that it produces a bunch of different colors. Kind of like its cool party trick here is it turns this like really blood red pigment on our McConkey auger. And I will tell you, this is more of like a party trick than it is like a true identification, just because it happens relatively infrequently for the bug. So it happens around 40% of the time, not necessarily all the time. And I'm
just taking the dye from the auger.
It's actually doing, if I remember correctly, it's doing like some sort of decarboxylation reaction where it's putting that product out on the outside.
Okay.
But yeah. And then this here, it's kind of like can appear like a goldish sort of color, occasionally look red on there as well. But that's our Serratia. It's an opportunistic pathogen, and it's associated with nosocomial infection. So when I say nosocomial, you guys know what that means? Okay, good. Okay. I don't want to like be throwing out fancy words and you're just like, what the hell is he talking about, dude? Yeah. And this was like, when I was putting this together a couple years ago, I was trying to find out something interesting about Serratia. And it's the 12th most commonly isolated organism for pediatric patients in North America, Latin America, and Europe. So that's like super specific. But you won't need to know like that at all. So the production of the red pigment, it's motile. It's a slow lactose fermenter, so usually first day it's not going to be that pink color, but the second day it will be. It's DNase positive and ONPG positive as well. The other thing I will add to this that's not on this slide, it is gelatinase positive as well. So when we do a gelatinase test, we'll kind of, hopefully I think I've got a Serratia in there and I'll kind of show you guys what a gelatinase positive looks like. And it can be passed from person to person, usually through, but we can usually get it through urinary catheters, respirators, IV fluids, and other medical solutions, and it can survive harsh environments. The really crazy thing about Serratia, have you guys ever heard of Operation Sea Spray? Oh man, we're going to dump down the world of like, well, it's not really a conspiracy theory because it's proven true. But the U.S. government did a bioweapon experiment on the city of San Francisco. Yeah, and so they released Serratia marcescens and another bacillus species out into the environment. And ultimately, they just wanted to see how it would spread through a population. And the only reason they ever found out about it is because a bunch of people presented with a bunch of weird UTIs with a bunch of red pigmented organisms.
So the lab was like,
something's weird. Yeah. So this was something that was done by our U.S. government. Luckily, this thing isn't like super deadly, but another example of.
Were they trying to use it as an example for like something more deadly, like anthrax or something? Yeah. Okay. Cool.
They just want to see how it would work in like a densely populated area.
Oh, cool. Yeah. Wait, do you know the exact number of like how many people who got it?
Not exactly, and you can Google it. It's there's a Wikipedia page for it. I know that for a fact.
Do you have a year?
I want to say it was like in the 70s, 60s or 70s. But Operation Sea Spray is what it was called. So, Serratia. Cool. Next bug here, Hafnia. Yeah, it kind of looks cool. It's kind of like spread out. Almost looks like it's got like a web-like thing.
Yeah, I was gonna say it kind of looks like.
Yeah, it kind of does look like ice. Yeah. But this is what it looks like on the plate. It spreads out, and this is usually after a day or two of incubation, not necessarily like right away. And then on McConkey auger, it's a non-lactose fermenter. Also, these ones smell pretty great. Kind of smells like garbage. Say that again.
Worse than bile?
I would say they're like bile has like a sharp smell to this. This one just kind of smells like garbage. But the big one from this is going to be Hafnia alvei, formerly an Enterobacter species. I just throw that in for historical context. It's mostly associated with GI infections, and I'll tell you a lot of times we overlook it in our stool cultures because normally it's going to be mixed in with a whole bunch of other stuff. And it's usually seen with other pathogens, so like it's very rarely by itself, and it's commonly seen in immunocompromised patients. So that's usually where we see them, is catheter specimens from somebody who's been in the hospital for a long time, maybe for a kidney transplant or something like that.
Is it typically like flora?
Not usually. Not usually, especially in that sort of situation. So here's a comparison of all the bugs that we've talked about so far here with Enterobacter rogenes, Enterobacter cloacae, Hafnia, both the klebs, Pantuia, and Serratia, and kind of showing you some significant biochems that you'll want to keep track of for each of them. Okay.
If we remember this thing, will that be that chart basically?
I will tell you, there are some questions where the chart will be your sole answer, where you'll be able to like follow it all the way down for the biochem and you're good. But there will be some times when you can kind of use the chart to rule things out, and these charts will be helpful to fill in the blanks. Okay, so keep that in mind. Cool. Sorry.
I was trying to see if there's a shortcut.
Not really for this one, I'm sorry. Yes. Remember everything for the rest of your life.
Yes.
Just, yes, literally just scoop it up and put it in your brain. Yes.
Driving.
If only osmosis worked, but like knowledge.
I mean, have you tried it?
No.
I slept with my notebook last night.
Didn't work?
No.
Damn. Well, try harder then. I don't know. Okay, Salmonella species here. So on blood agar, it just kind of looks like a regular old E. coli with a flat gray colony, nothing really super unique about it. On McConkey agar here, this is a up close picture of like what they look like. So the non-lactose fermenter is going to be that Salmonella species, and the lactose fermenter is going to be like an E. coli there. And then super cool on our HE agar, this is one of those that will reduce our H2S. So it's going to turn black on there.
And why is it lighter in other corners, like, too? Is there any reason for that?
You talking about like,
It's not completely blue everywhere.
oh, it could just be the way that the picture was taken,
Right.
where the light's kind of coming off of it. That's one of the, so I'm going to digress from this lecture just a little bit. Just, it's one of the reasons why automated systems haven't caught on super well in biology, is because of this like color
Right.
discoordination. But now they've got AI that learns, which is really cool.
This is going to be slightly maybe a personal question. Have you ever seen somebody who's colorblind work in a lab?
Yes.
Okay.
My chemistry professor was, as a PhD, was completely colorblind, and he said looking at protein structures was a nightmare.
Right. Okay.
So, and there's definitely been people who do it here too. It's harder as a microbiologist, but
Right.
the way they've described it is like you tell them that's yellow and they accept that that's yellow, even though it looks like something else to them. It's possible. Okay, Salmonella. Something I want you to know about this is that infections are zoonotic, meaning they jump from from animals to humans. And I will tell you, all great pandemics start that way.
Really?
Yes. Here we are. It causes gastroenteritis, otherwise known as salmonellosis. Salmonellosis. And infections are generally caused by ingestion of fecally contaminated food. When you think about it, that's like super gross, but that's how we usually get it. And it's not really a danger to like people kind of our age, but as you get older, or if you're like super young, that's where the danger is usually at. And usually with these, we recommend that they don't use antibiotics to treat just because it can prolong the carrier state of that bug, unless it's absolutely necessary, i.e. move to a septicemia or something. And there's really two major types. There's non-typhoidal and typhoidal salmonella. I always like, you know when you like say a word too many times in a row, it starts to sound funny to you?
Yeah.
We're about to go down that path, okay? Oh, salmonella naming system. So the big species that we usually are talking about when we talk about salmonella, there are others out in the environment, but Salmonella enterica is the genus and the species name. However, there are over 2,500 different serotypes associated with them, and you're responsible for knowing them all. So
Perfect.
good luck. But the way that we're going to do this for nomenclature is naming is based off of serotype, virulence, package, and location. So their different names kind of have different connotations to them. But the way we're going to put it is Salmonella enterica, subspecies Arizoniae. So from that, I can tell you that it's a Salmonella species that probably originated in Arizona. Sometimes they're named after people, sometimes it's named after patient zero, sometimes it's named after a virulence package that's associated with them. That's just kind of how the nomenclature goes. And then we can shorten this even further. We can say Salmonella arizoniae. The enterica is kind of understood. That makes sense. So if you're reading ever a research paper about Salmonella or reading in your textbook, this is why they're abbreviating things the way that they are. Okay, so non-typhoidal Salmonella, this is what we think of most times when we talk about diarrheal disease. These are the ones where the disease, the onset of symptoms is usually 6 to 72 hours after exposure and typically lasts about 12 to 36 hours after that. And the illness can last a total of 2 to 7 days, kind of depending on your immune status and a whole bunch of other things. And treatment typically consists of electrolyte replacement therapy. And like I said, we don't want to use antibiotics because in this case, we're just going to prolong you holding on to that bug. Okay. Typhoidal Salmonella. The big one that comes out of this is Salmonella typhimurium. So this is Salmonella enterica subspecies typhimurium. Okay. And it's the thing that causes typhoid fever. Have you guys ever heard of Typhoid Mary? You heard her story? Believe it or not, a Kansas resident. Yeah. So she was a carrier of Salmonella typhimurium, and she was a food prep person. Worked for the United States government, worked in for the military and a whole bunch of other places. But what they were finding is wherever she went, and this is back in a time when hand hygiene wasn't the greatest, wherever she went, these people would come down with typhoid fever, and they couldn't ever figure out why until they traced it back to one person who was prepping their food and they were ingesting it. And so ultimately, what ended up happening is the woman ended up getting arrested for intentional food poisoning. And that happened a series of times, and she always went back into the same industry doing the same thing. She was just kind of she was a super spreader, if you want to think of it that way. So it's kind of interesting.
But she knew that she was hurting people? Okay.
She knew about it. Once they figured out that she was probably the cause, they figured out that she was a carrier for the Salmonella, and they told her about it, but she didn't ever want to give it up. So,
Did not care about other people.
did not care. Nope. She just wanted to keep doing her job. So I don't know. I don't get it. Crazy people. So difference between typhoidal versus non-typhoidal. Typhoidal has the ability to go systemic, meaning it has the ability to go from your GI tract into your lymphatic system and cause a systemic salmonellosis type of infection. Okay, so annually there are about 60 million cases of typhoid fever, and about 500,000 of them result in death. I will tell you, here in the United States, it is not as much of an issue as it is in different places of Africa. In fact, this is one of the things that they actively screen for routinely in labs out in Africa. So, and the treatment for this is actually pretty cool. I don't ever talk about this in here. So they use a drug called ciprofloxacin. Have you guys ever heard of ciprofloxacin? It's a drug that's a fluoroquinolone, and we'll talk about this, what a fluoroquinolone is. But the cool thing about this, so Salmonella likes to get inside of your macrophages, inside of your GI tract, and that's how it hitches its ride onto your lymphatic system and causes that systemic infection. What ciprofloxacin does really well is it concentrates in high concentrations inside of those macrophages and ultimately prevents that infection from progressing any more than just a GI infection. So that's why it's the treatment of choice.
So once it gets into the macrophage, it just basically kind of conceals itself.
Mhm.
Gotcha.
Yeah, your body just walls it off with that drug. So pretty cool. Also with typhoidal salmonella, the way we know that it's typhoidal salmonella is the presence of this VI antigen. So remember, this is one of our capsular antigens. One of the ways that it's also able to evade our immune system is with the VI antigen. But this is how we ultimately call it a typhoidal salmonella. Kind of the progression of typhoid fever here. So week 1 is usually your septicemia, and then week 2 is usually when we find it in your stool, which is kind of crazy to think about. Like you're already kind of screwed at week 1, and we won't even be able to detect until week 2. Then week 3 is usually the place where you're going to experience the really high fevers and any sort of complications after that. And then week 4 is convalescence. So you either get really, really, really bad and potentially die from it, or things get better for you. So it's about a month-long disease process.
So the septicemia won't kill them in the first week?
Not usually. Can if it goes untreated, which septicemia is extremely deadly. So there's a statistic floating around that for every hour that you are not on the correct antimicrobial, your chance of mortality goes up 8%. So depending on what part you are in that septicemia, will kind of tell you your survival chance. So even after 24 hours, you're already looking at about a 70+% mortality rate. That's why it's important to go to the doctor. Cool. All right, great growth characteristics of Salmonella. We see these quite frequently in the lab, so you guys will see them and kind of be able to recognize them. But it's a non-lactose fermenter on McConkey and on our HE agar. It's got black centers on our HE agar here. So this is something boards will like intentionally point out to you, is the fact that it's an H2S producer, but also the fact that the centers of the colonies themselves are black. This is something that's indicative of Salmonella typhimurium. So they might be like, what's the potential pathogen here? And give you a whole bunch of other H2S producers, but the fact that they pointed out that the center was black is important.
So that's just specific for the type of bacteria.
Correct.
Okay.
Yeah. Okay. So on top of that HE agar, another thing we can use to just kind of help eliminate the noise in the culture and the other things that might be growing there is the use of this stuff called Salmonella and Shigella agar. And this is what it looks like on here. And if you could put money on it, this Salmonella is also black, and the Shigella is kind of like a clear colony, and a lot of times they'll even appear like red on the agar itself. And then also a Salmonella, it's able to grow on citrate. So the important thing about citrate is it is a very basic carbon source. There's not much to the molecular structure of citrate. So if it's able to utilize that, it's pretty used to living in pretty harsh environments. So.
And the citrate is the one that goes from, is it green to blue?
Exactly. Exactly.
What else can grow on citrate?
Citrobacter is another one. Pseudomonas is another. Things that are usually kind of happy with really crappy environments. So you can kind of think about that. Cool. Questions about Salmonella? That's a fun bug topic. Okay. Proteus species. This one, kind of gross. This one, if you look at it here, it kind of has like the effect here, and you kind of see the center of the colony right here. So this is what we call a swarming Proteus. Okay, so you'll hear them say, oh, something swarmed the plate. This is what we're talking about. Okay, it just takes over the entire thing. Yes.
We went over this previously. What is the thing that inhibits Proteus?
Pseudomonas can do it. I want to say staph.
Yeah.
Oh, you're talking about the agar?
It's like the specific chemical or protein or whatever that's in an agar that the PEA.
Mhm. It's just alcohol. Alcohol will inhibit it. Yeah. Alcohol will inhibit Proteus. Yeah. But this is the most common case. There are Proteus that exist that don't actually swarm like this, but this is common. McConkey agar, which again, they kind of break the rule with this because we say that all Enterobacteriaceae grow on both blood and McConkey agar. Some of them don't actually grow on McConkey agar, but when they do, they are non-lactose fermenters. Also with Proteus, it's got a smell associated with it. No, it smells like hot garbage in my opinion, but they say it smells like burnt chocolate.
Who?
Burnt chocolate.
Who says that?
Crazy people, that's it.
Oh, okay. That's dark.
Burnt chocolate. So if they mention that in like a case study and they say, oh, it smells like burnt chocolate, that's what
That's
it's.
like an official description.
That is the official description of
You
it.
said it was non-lactose or lactose?
Non-lactose.
It smells like hot garbage or burnt chocolate.
Yeah. Hot garbage isn't official, but the burnt chocolate is. But there's two species that we really care about. There's Proteus mirabilis and Proteus vulgaris. I will tell you, mirabilis is more like an E. coli in the sense that it's considered normal. Vulgaris is kind of more of an asshole. So it's more antibiotic resistant. It's also more virulent. Both of these bugs have a very, very strong urease enzyme. So if they are, the crazy thing about Proteus is the fact that it's hypermodal. So you see that kind of here on the plate where it's just kind of growing over everything. But with that and the combination of a really strong urease enzyme, it's able to progress up your ureters and into your kidneys really, really quickly if it's left untreated. And when it does that, it can kind of break down that urea and produce these magnesium salts that form in your kidneys, and you get kidney stones from it. So add insult to injury, you've got a UTI, and then you'll have kidney stones with it as well, potentially. So just be aware of that.
What would you use to get rid of that? What medication?
They can give you like a beta-lactam is usually what they go with, or like a fluoroquinolone. Those are pretty easy to take, but there's plenty of options out there. Yeah, primary pathogen in the urinary tract, but we can find it in a bunch of places. We get it in wound cultures and things like that too. But it's also normal flora for the GI tract, and it's considered a contaminant in urine cultures sometimes. So occasionally we might get a Proteus that swarms over a pure culture of E. coli. We'll ignore that Proteus and know that the E. coli is the actual pathogen here. So cool. So for some identification things, I told you it smells like burnt chocolate. It smells gross. It's also an H2S producer, so it'll appear like a Salmonella species on that HE. So keep in mind, remember, this is a GI colonizer. So if we're looking at a stool culture and we see H2S, we want to confirm that it's actually a Salmonella before we do anything like that. So these are going to be the two ways that we will, or a good way of kind of like speciating them out. I will tell you, your go-to for Proteus mirabilis versus vulgaris is going to be that indole reaction. Indole negative is Proteus mirabilis, indole positive is the vulgaris, and it's really a vulgaris group. But I will tell you that's going to be your go-to there. Questions on that? Okay. My friend Citrobacter here. So this one on the plate will look identical to an E. coli. Some of them are beta hemolytic, some of them are non-hemolytic. They will be lactose fermenters on here, and you'll see kind of like a bile precipitant forming around them. The big thing that's going to kill you with Citrobacter is it smells like poop. I give it the nickname Citrobacter for a reason, because it literally smells like feces. This is the thing that gives poop the poop smell. Okay. So the two species we're going to consider here are Citrobacter coasteri and Freundii. Both are inhabitants of the GI tract, but they're most common in UTIs, but we do occasionally get them in septicemias, meningitis, and brain abscesses. These ones are also H2S producers. And the thing, they get their name Citrobacter because they're able to utilize that citrate as a sole carbon source. They don't need a whole bunch. And hopefully I have one of those on a plate. You guys can see what I mean. It literally smells like poop. So far we've talked about.
I think I can believe you on that. Yeah.
That's okay. No. You guys have to suffer too. Okay? These are the three bugs that we've talked about so far. Citrobacter coasteri, I'm sorry, I forgot to mention this, is not an H2S producer. It's just Freundii that is, but these are going to be, if you're going to compare H2S producers, this is what I will look at here.
The coasteri is not.
Correct. It is not a H2S producer.
Would that be on a chest possibly?
Potentially.
Yeah.
Potentially. Or part of it. Can you do that, please? Okay. Edwardsiella tarda. What a name. I think I've only seen one of these like ever, and I can't even remember what the source was on it. But it looks like regular gram-negative flat gray kind of nothing really discrete or descriptive about it. The one that I have seen is not actually a lactose fermenter. It was a non-lactose fermenter. So it can be either, but it's an opportunistic pathogen that can infect many sites. It's usually associated with gastroenteritis. We get it occasionally, and now that I'm thinking about this, it might have been a wound care patient that we got it from, but wounds, systemic infections for people with underlying diseases, you guys will learn anybody that's immunocompromised or any sort of issues other than just the infection, they can get a whole bunch of other things with them. But this is also an H2S producer. It's also indole positive, which are the things I would remember about it.
Is it found naturally in fresh water?
Yep. You got it.
So if you have like an open wound and you go into water that has it, you can probably.
Amongst other things, yes. By the way, you guys, who wears contacts here? Oh, we're gonna ruin your. Okay, we're gonna get the parasites. I'm sorry.
Oh my.
I'm not even gonna talk about it here. But
Wait, would you recommend that we should be doing daily contacts
Yes.
instead of, okay.
don't sit them in solution. Don't put them in solution.
I don't worry.
Contacts.
I actually, no, I think
Have to put it in
true,
the solution.
because a little bit of
Daily
T1,
ones you don't.
but like if I leave it
Like,
in
you don't have any
the solution, say I don't change the solution every day, I would get conjunctivitis.
Yeah. Yeah.
Interesting.
Does anybody has anybody ever used a neti pot?
Yeah.
Is that the?
I've never
used it, but I want.
Don't use-
I suspect a free living amoeba in your brain at some
Go
point.
for it.
So,
Eat all the brains.
but yeah. I would remember this chart right here mostly for the list of H2S producers. So part of this review, I'll do this review with you guys too before the exam, so I can kind of help lead you in a direction. But something that's not as information heavy. But I will ask you, what are the 5 H2S producers, and I will expect you to tell me these. So kind of know this list right here, okay? Also with this, there are not very many H2S producers that they will call out on the exam. So if they say it's H2S positive, you can pretty much come down to this list of 5 right here. Okay.
And then they say it's like indole positive,
Mhm.
and then you're like, okay, it's between these two.
Yep.
Got you.
You got it. I listen.
So wait, question.
Yes.
So they send in like yearly, these just random things that you have to be able to test and figure out.
Mhm.
So that sucks.
It does.
So how long does it usually take you to figure out these unknowns?
Not that long. It's usually about the same as the patient. So if it's
Do they ever send weird stuff or is it like normal stuff?
sometimes they'll send weird things. The weird thing, okay, so when it comes to CAP and like weird stuff, it's
Right.
usually in the context of a negative result.
Sure.
So if they send us a negative result, we're like, what did we do wrong? Like something's weird here. They very seldomly send us a bug that we just can't ID. So, so happens occasionally, but not usually. And if we do answer it wrong, they be sure to let us know about it. So, okay, Yersinia. Let's talk about Yersinia. So, by the way, when I picked up this lecture, this whole thing for Yersinia was like 20 slides long. So now we're down to like 5, if that. So
Is it because it's less important?
the guy who had it before me was like a super Enterobacteriaceae nerd more than I am, and he like Yersinia. So Yersinia pestis is, if you heard that before, of that. Yeah, like talking about the plague, and so he would nerd out about the plague with you guys for like a solid 30 minutes, and we're not going to do that. So, but this is what Yersinia looks like on blood auger here. It just kind of looks like this flat gray colony. Over here on McConkey auger has kind of that pink color. The big auger I want you guys to know here is the CIN auger. So Yersinia intercolitica is described as having like this bullseye pink sort of colony there, and that's going to be like one of the ways that we can definitively ID this bug. Okay, so CIN auger is what it's called. It's also called Yersinia selective too. You can see those terms kind of interchangeably.
I don't see a Yesenia select.
Thanks.
And you said that
Mhm.
bullseye peak colony can be a definitive item for you.
Can be, yes. So there's three main types of Yersinia that we're going to talk about. There's Yersinia intercolitica, there's Yersinia pseudotuberculosis, and Yersinia pestis. All of them can cause fever, abdominal pain, and diarrhea. And oftentimes, at least this Yersinia intercolitica can be confused with appendicitis. So before you come into the ER saying, I've got appendicitis, sometimes they may want to rule out a stool pathogen as cause. So Yersinia intercolitica, the source is typically undercooked or improperly cooked pork, which you guys will find out. I'm going to ruin food for you guys, and I'm sorry. Unpasteurized milk, so don't drink unpasteurized milk. And they can survive and multiply at 4 degrees Celsius. They really like room temperature. They even like this cold temperature too. But it's usually associated with GI tract infection. And it's kind of weird, like with this bug right here, if we were ever to get a request from a doctor to culture for this, we would leave our auger out on the bench top to see if it grows. So Yersinia pseudotuberculosis is similar to intercolitica, except you can kind of figure out from the name what type of infections it grows or causes, but respiratory infections are typically what we associate that with. Cool. So there's another picture of that CIN auger. And something about Yersinia that I want you guys, that we mentioned very, very early on in this lecture, is the motility and the temperatures that it occurs at. So Yersinia intercolitica will be motile at room temperature or 25 degrees Celsius, same with tuberculosis, but won't be motile at 37. And then Yersinia pestis doesn't like to be motile that often. And really this motility at 25 degrees is more like a variable thing for them. Okay. And then Yersinia pestis, our friend, the cause of the bubonic plague or the Black Death. And really we've traced the cause to being bitten by a flea that has been infected off of feeding off of a dead rodent, which is kind of gross. And something like 10 to 20 cases occur in the United States annually, and about 1,000 to 3,000 worldwide. So kind of interesting about this is one of those that they'll give you like a case study for it. And they will tell you this is endemic in the southwestern part of the United States. So they'll be like gunning for it real specific. They'll be like, okay, there's a patient that presents with an infection that resides in Arizona or New Mexico, and they have developed swollen lymph nodes with a fever, and the bug kind of looks like this. They're going to be gunning for Yersinia pestis. So if they give you anything for the southwestern part of the United States, just automatically start thinking this. Okay. And symptoms usually occur 2 to 6 days after exposure, and they typically include like swollen lymph glands, fever, chills, extreme exhaustion, coughing, and expelling bloody sputum. Have you guys ever seen, are you guys a fan of House, the show House? Have you ever seen that? Okay. Has anybody seen, have you seen the episode about the patient who has the bubonic plague? Yeah. Okay, so the reason it gets the name bubonic plague, what those swollen lymph nodes are called, they're called buboes. And what they end up filling with is like necrotic fluid inside of there. So when a doctor goes in there with a syringe, occasionally they'll pull out a fluid that appears black or kind of like bloody at sometimes. And in Dr. House, I was going to say that's like pretty much exactly how that would present to somebody, is these kind of this black lymph node fluid. So that's usually one of the telling signs that we got some bullshit going on. Okay. And if you guys were ever kind of like interested in some like great plagues throughout our time as humanity, this is a few of them. There is far more than just this. And currently, oh, I don't know if it's currently, because COVID kind of messed up the flow of things. But it's like every 2 years, Madagascar experiences an outbreak of Yersinia pestis. And has anybody ever played the game Plague Inc.? Everybody knows in that game, the best place to start plague is Madagascar. So we're just waiting for Yersinia pestis to take over. But so
Is it similar to risk?
say that again.
Is it similar to risk?
Similar, but with bacteria.
So with the Yersinia pestis like going on right now in Madagascar.
I don't know if it is this year. I think this would have been the year that it would have been due for it. It's like every even year or something like that. But it usually has like an outbreak of like a couple thousand cases and then just kind of disappears. It's really weird. So here's a good way to, or a good chart to kind of differentiate between them. But with this one, a case study is usually going to be the way that they're going to present it to you, and they'll give you something real specific. All right, Providencia. So this is what it looks like on blood auger. I couldn't find a really good picture of it on McConkey auger, but it's a non-lactose fermenter on there. To me, the way I would describe it is it looks like a non-swarming Proteus. It almost kind of smells like a Proteus too. It was also formerly a Proteus species, so it kind of makes sense, right? So there's 3 organisms that we want to know about. Providencia, yes.
I think last year.
What was it last year? Okay.
August 29th. August 30th, 25.
Heck yeah. I wouldn't lie to you guys. See? Yeah, so every couple of years it comes up. Still a problem. Okay, so Providencia, the 3 species we're worried about is Providencia returgeri, Stewardii, and alkalicophacians, because you can swallow your tongue when you say a word. That's the microbiologist way. Typically, we're going to see these as UTI things, so we'll occasionally get them in like wound cultures, but mostly UTI specimens. And they possess a urease enzyme, kind of like other Proteus do. It's also associated with long-term indwelling catheters. And alkalicophacians is associated with gastroenteritis. And then, like I mentioned, they're not lactose fermenters. But this is going to be what you're going to have on here. And I'm trying to think. This will be one of those where, let me think on this one. The way they would be, I would stick to your chart on this one as far as like an identification goes. I wouldn't necessarily go with this chart right here. I'd follow your little chart that I gave you
So
guys.
you don't think that we should look at this chart at all?
I'm trying to think of anything that would be specific to it, but this one's not really. Let's just like to take it further. Morganella morgani, also kind of like a Providencia. The way they both look very similar on the plate. They're like a non-swarming Proteus. And kind of something that's unique to Morganella is the fact that it kind of turns this auger like kind of a darker, deeper red or almost a black color underneath it. I wouldn't necessarily call it hemolysis, but it's just kind of like this weird phenomenon that happens underneath it. It's a non-lactose fermenter on McConkey auger. Formerly a Proteus species, so that kind of makes sense. It's an opportunistic pathogen, often associated with UTIs, and also possesses a strong urease enzyme. And as far as like anything else goes with that, just leave it at that. Okay. And I would stick to your chart for IDs on this one as well. Okay. Plesiomonas shigaloides. It's the next one we're going to talk about here. Plesiomonas. Yeah, it does sound like a dinosaur. So it's kind of just looks like a regular old gram-negative rod, non-lactose fermenter over here. The big thing I want you to remember about Plesiomonas is that it is the only member of Enterobacteriaceae that is oxidase positive. And that's going to be usually your tell right there. Everything else will rule in as Enterobacteriaceae, and then they'll be like, oh, and it's oxidase positive. But it got its name because it cross-reacts antigenically with Shigella. So if we were to do like a serotyping with them, we might be able to confuse the two. But it's found in animals, freshwater, and soil. Found in the tropics, subtropics, and Japan, which there's some cool things that come out of there that are dangerous. And it causes gastroenteritis, and like I mentioned, it's a non-lactose fermenter. So cool. Those are all of our organisms. Questions on any of that stuff? Yeah, just remember all of it. It's fine.
I feel like I was asking questions, right?
Yeah. I'm sure you guys will come up with a bunch of stuff. I will tell you, I modeled this entire PowerPoint our objectives. So there's going to be a lot of things that you'll be able to answer directly from just this PowerPoint. So
just memorize the PowerPoint.
basically, yes.
Cool.
Yeah. And I want you guys to recite it verbatim to me on everything that's on it. Okay, so let's talk about the fun part here, which is some other identification methods. So something we're going to do today are the API 20E strips, which did you guys ever do API strips at college or anything like that? Okay, awesome. This will be the first time for you guys. Like we did it. But it is a rapid biochemical ID. It's just one long strip. They look like this.
Oh, yeah.
Yeah. So we'll kind of walk through that today. This will take a minute for us to set up, but we'll get that going and we'll take a look at them tomorrow and do the interpretations. But based off of the biochem identifications that are occurring here, we'll give the bugs a score and we'll be able to plug them into a computer system and that will tell us an identification. Cool. The other identification method that we have are these nucleic acid-based things. So we were kind of mentioning about the other E. coli species that we can't really culture for. We have to do PCR. So this is an example of what our gastroenteritis panel looks like. We're going to do PCR for rapid identification. So we test all 22 of these targets in about an hour. So that's kind of the rapid way of doing this. And it's a cultureless system. The other method that we use a lot of that you guys will get plenty of practice on is MALDI-TOF. We just call it MALDI. Some people call it the Brooker. There's another version called the Shumatsu. There's a Vitek. There's all sorts of wonderful different names for it. But this is what it is. And we'll talk about the technology when you guys get into section 2. But essentially what happens is we've got a little plate with a little well on there. We put a little bit of an inoculum onto our little wells here, which is what's down here. And then we overlay it with what we call a matrix. And the matrix, the sole purpose of it is to provide a charge and stabilize the proteins inside of that organism. And what we do is we put it inside of this mass spec instrument where we then blast it with laser and then we hit this detector. And based off the time that it takes to hit that detector determines the mass of the proteins on there, puts it onto a little plot like you guys are used to seeing for mass spec, and then it tells us what the identification is. And it all happens in about 45 seconds. Pretty cool. This is the best way to do it. When do you guys have antimicrobials? Like next week? Is that what it is?
Next Tuesday.
Okay. I want to show you guys like a cool video that will kind of like, I don't know. Oops. Maybe be cool to you guys. So I'll kind of explain it here. It applies to this lecture and then it applies, yes, I'm not a robot, thank you. Traffic lights, there we go. Okay.
I did it once, but it took a while.
So this is cool for multiple reasons, other than Matt's a nerd. This is E. coli growing on a petri dish, and I'll kind of talk to you about, kind of explains the evolution of antimicrobial resistance within that species over the course of a week. And it kind of emphasizes the importance of choosing the correct drug at the right time, making sure that we are listening to the lab results that we're putting out. If we're saying something's resistant, we need to act on it accordingly. So something came out of Harvard Medical.
Building was basically fish. Except it's 2 feet by 4 feet. Set it up. That's not bad. Yeah, basically bad. But it's exotic side. No exotic. Just if you 5 out of that. I'm still up. Top of it. Dagger. Period. That black continent. Period. White. First, it's biotic. Got mute. Right. Then got it. Starts. Found it. You have by process of cumulative sedimentation that bacteria is biotic and
So that's just your regular friendly E. coli. That's not even like things like Pseudomonas will develop resistance in the span of a day. So like that. So it's a big deal. But any questions about any of that stuff? Okay. I want to give you guys like a mental break for like, you guys want to do like 30 minutes? It's like lunch-ish time. You guys do lunch? Is that like a thing?
Well, we like here, whatever.
Okay. Well, I would expect today that we're going to be here for a minute. So I would say definitely go eat